All right, everyone. Welcome to the lab exam review. This is for lab exam 1. And to prepare for this exam, you are going to want to review the PowerPoints and any associated handouts for each lab. I made content videos and posted some lab reports. You can see some questions that you may want to know the answer to. And then, of course, there is the PowerPoint study guide that is posted that you will want to go through and complete in order to make sure you're ready for the exam. So this Kahoot is going to be posted, but I wanted to make a video to walk through the answers. To speed things up, I'm not actually going to let the full 20 seconds go by, and to be honest, I may just click something at random, so we'll see. But here we go. All right, so let me show the media. There are going to be quite a few pictures on this exam. So you definitely want to know the different types of media. So this is a test tube full of solid media. And so we call this an agar deep. Okay, an agar plate is solid media in a Petri dish. A slant is solid media remember, agar is liquid at really high temperatures. And so when we pour the sterile, really hot media into a test tube to prepare the media, we then tilt it at an angle so that as it cools, when the agar solidifies, you'll have a nice slant. So a slant is literally slanted. And then the broth is the only type of media that does not contain agar. It is a liquid, and it has like a goldish color. And remember, you can see through it, okay? It is not naturally turbid. All right, next question. Look at me winning. Go me. All right, so the media shown in this picture is an agar plate, hence the Petri dish. And remember that agar is a solidifying agent. That is it, okay? It offers no nutritional value for the microorganism. And that's a really good thing because remember, Hesse is the woman who suggested we use agar in media because previously they'd been using gelatin, and that was able to be used as a source of nutrition for the microbes. So they were metabolizing and breaking down the media. So things weren't growing on it because the organisms were eating it. So agar is strictly a solidifying agent. Okay, but remember, like I said in the previous question, it is a liquid at really high temperatures, but as it cools down, like to room temperature, it will solidify. All right, next question. All right, so for questions like this, you want to make sure you know how to describe a colony. But remember, there are different categories. There's shape, there's margin, there's elevation. And so there are certain terms associated with each of those categories. So you want to make sure that you know which terms are associated with margin, which terms are associated with describing elevation, okay? And so for the colony that was shown here, you always want to look at like an isolated colony. So like this one right over here is a good one, right? And overall, it's round, okay? If it was irregular, well, it would just have a crazy shape. Lobate is when it looks like it has very curved ends, like little lobes at the end. And then that's all I wanted to say. So let me keep going. But make sure you know those terms, okay, y'all? Oh, here's another one. Ooh, there's a lobate. All right, so let's take a look at it. So remember, the margin is you're only looking at the edges, okay? Not the overall shape. You're just looking at what's going on at the edges. And see how they have these nice round edges, right? That is going to be lobate. All right, next question. Maybe. There we go. All right, so remember there are 3 different ways you can describe growth in a broth. The growth in this example is up at the tippity top. It's only at the top. So we call that pellicle growth. If the growth had only been at the bottom, it looks like a little clump at the bottom, then we call that sediment. If there's turbidity or growth all throughout the broth, then it's uniform turbidity. We also call that just turbid. We also call it uniform fine turbidity, but turbid is there in the description. All right. So for this question, remember the counter stain is the last stain that is added. So in this case, it is safranin, okay? So when it comes to the stains, you are going to want to know what is the purpose of the stain? Why do we do it? What information are we trying to glean from this stain? What stains are involved and why? Right? A lot of them are a lot of the stain, can't speak. A lot of the stains selected are there for a reason. Okay, so you want to know why each stain is used. And then of course, how to interpret the results. So if we look at this picture again, you'll see a mix of red and purple cells. So the only 2 stains used in the gram stain is crystal violet, which is purple, and safranin, which is red. So gram positive is one color, gram negative is the other color, okay? For the gram stain, it's a very important stain. It differentiates bacteria based on their cell wall, and their cell wall is what allows this stain to work. So remember, the primary stain is crystal violet. It is a basic stain, so it's positively charged. So it's going to be attracted to all bacterial cells, right? Because all bacterial cells have a negative charge. So initially, when you add the crystal violet, both gram-positive and gram-negative organisms will be purple. Next, you add iodine, which is a mordant, and so it is going to essentially like link arms with crystal violet, making it a much larger molecule. And then, so again, after adding the mordant, both types of cells are still purple. But step 3, when we add the 70% ethanol, the decolorizer, that is the differential step. And that's because this is when the cells appear different under the microscope. For the gram positives, they have really thick cell wall. Very thick layer of peptidoglycan. And so when you add the crystal violet and the iodine, it gets in there nice and deep into this cell wall. And so when you add the decolorizer, the alcohol dehydrates that thick peptidoglycan. So it goes and it kind of like shrivels up a little bit and it traps that large crystal violet iodine complex inside. So that crystal violet cannot leave. Those cells are forever purple. However, for gram negatives, which have very little peptidoglycan, so it's not really trapping in that crystal violet. Remember, though, it also has an outer membrane. So the crystal violet is there between the peptidoglycan and the outer membrane. But when you add alcohol to a phospholipid bilayer, which is the outer membrane, it dissolves it. So with the outer membrane dissolved, the decolorizer causes the crystal violet to be washed away. So at this point, under the microscope, gram positives are purple and gram negatives are clear. Okay, but clear cells don't do us any good, right? You want to be able to see them under the microscope. And so we always add a counterstain, which in this case is safranin, which is red. And so when you add the safranin, it's going to be attracted to the bacterial cells and the gram negatives will appear red. All right, next question. So this is going to be true because gram-positive is purple, gram-negative cells stain red. So for this one, think to yourself, well, what is the morphology of these cells? And you would say these are cocci because they're circular. And if I asked you what's the arrangement, you would say staphylo. You have to look and see collectively what's going on. Okay, and we normally see them all in clusters. There is a little diplo over here, but look at all of these staphylos, right? Mostly they're all in nice, pretty clumps. Yes, you have to kind of look collectively to see what's going on. But you have to make sure that your specimen doesn't just have way too many cells, because then everything looks staphylo even though it's not. Now, I will not give you a picture of too many crowded cells, but just in the real world, that is something you have to be careful about when you stain something, because if you have too many cells, it's going to be hard to stain properly, but then they always look like they're in a staphylo arrangement. But luckily, we know that bacillus cells cannot have a staphylo arrangement because they cannot divide along their long axis. So you should never have a staphylo-bacillus arrangement. All right. So the acid-fast stain is a structural stain. It is specifically looking for organisms that have mycolic acid in their cell wall. And the only organism that has mycolic acid is Mycobacterium. And so this stain, because Mycobacterium has a waxy layer, waxes are lipids, so we need a lipid-soluble stain here, and that's why we use carbofusion. So carbofusion is the primary stain because it can penetrate through that waxy layer. So a fuchsia color is going to be positive for the acid-fast stain. So for this one, remember that endospores do not look or act like regular cells, okay? These are very hardy forms, and it's hard for most stains to actually penetrate through and stain them. And so we have to use a special stain, malachite green, which has a low affinity for cellular material and it's water soluble. And so because endospores contain keratin, they're, like I said, very hard to penetrate. And so what happens is there are two approaches to staining endospores. The Ziel-Nielsen method, which is the hot method, is where you essentially create really hot steam and you set your slide over that steam, and then you add the malachite green and you wait about 5-ish minutes. And the steam is going to help, you know what? That is not the Ziel-Nielsen method. Sorry, the Ziel-Nielsen method has to do with the acid-fast stain. I apologize for that. But with endospore stain, you can still use steam, and then you just add the malachite green and you let it sit for a while. Or you can get a really concentrated version of malachite green, and you can just let it soak on your slide for a while. Okay, so again, I'm sorry, Ziel-Nielsen method is for a different stain. I apologize. But here we can still use steam, or we can use a concentrated solution to let it get through. Okay, sorry about that, y'all. All right, next up. Which stain cannot include heat fixing? So this is going to be the capsule stain, because capsules are a sugary layer, right? You don't want to subject sugars to heat, they'll disappear. But it can also cause those specific bacteria to kind of shrivel and be manipulated in such a way that you can get artificial halos when you shouldn't. So heat fixing can actually get rid of the capsules and give you a false negative for some species, and for others the heat just warps the organism and messes with the stain, and so you can actually get artificial halos when you really shouldn't, so you can get a false positive that way. Okay, so no heat fixing on the capsule stain and no heat fixing on the negative stain, because for that stain you're trying to be able to measure the length of the cells, or you are dealing with heat sensitive organisms like spirochetes. And so heat will completely warp them. So you don't want to heat fix for these two stains. All right, true or false? All right, so the iris diaphragm controls how much light passes through onto your slide. So it will help provide contrast when you're looking through the microscope, because maybe you stained a little too long and so you have very colorful cells. Well, you might need a nice bright background to see everything clearly. Or if perhaps the stain wasn't as potent or you didn't leave it on as long and you have like a really light color red, for instance, then sometimes you want to decrease the amount of light coming through to try and give more contrast. So the iris diaphragm controls how much light hits your specimen. The condenser is going to be what condenses light onto the specimen. So remember, I posted a video about the tour of a microscope where I walk through all of the parts of the microscope and their function. You are definitely going to need to know the parts of the microscope and what they do. You're going to want to know the name and magnification of each of the objective lenses. So the scanning lens is 4x, low power is 10x, high power is 40x, and the oil is 100x. And then you need to be able to calculate the total magnification. And so that's where you're multiplying the objective lens magnification times the ocular lens magnification. And all the oculars always have a 10x magnification. So here's where I'm testing. A, do you know how to find total magnification? And B, do you know what the high power lens is? So high power lens is 40x. Multiply that by the 10x ocular. 40 times 10 is 400. All right, so remember with the streak plate, our goal is to get isolated colonies. And as you can see in this picture, this person did not. And so there are really two reasons why someone may have gotten these results. One is that they didn't flame their loop in between quadrants. Because remember, we flame the loop in between quadrants so that we're killing any remaining cells on the loop, right? Because our goal is to decrease the number of cells that we have. So we're depositing single cells in that fourth quadrant. So if you don't flame in between quadrants, then you're just constantly putting lots and lots of cells in a very small space. The other potential thing that happened is after flaming their loop each time, they dipped their loop back into the broth, in which case they're putting thousands and thousands of cells back onto their loop when they streak that new quadrant, okay? You only use your broth once at the very beginning to introduce cells into quadrant 1. After that, you set it far, far away from you so you cannot be tempted to put any more cells on your plate. So make sure you are able to kind of figure out what went wrong on a streak plate. And there are examples in the lab report. Oh, I was waiting for y'all to answer, my bad. Basic dye stain has a positive charge. Why, yes, it does. Okay, so basic dyes have a positive charge and an acidic stain has a negative charge. And so because of this, a simple stain, which uses basic dyes, you're gonna have nice, brightly colored cells and a pretty white background, okay? Because the dye or stain is gonna be attracted to these cells. So the cells are gonna turn crystal violet or red if you're using safran, whatever it may be. And so the background is gonna be bright white. With an acidic stain, that means it's negatively charged. So the stain is going to repel the cell. So you're gonna get a nice, pretty dark background and bright white, colorless cells. So it's pretty neat looking, and you'll see pictures of those in a few minutes. Or right now. Is this a negative stain? So ask yourself, is the cell colorful or is the cell colorless? In this case, the cell is colorless. Pretty little spirochetes, so I should answer this question. Okay, so again, this is Congo red, which is making the background a reddish color and repelling the actual cell. So the cell looks colorless. And these are spirochetes, which is what you often use the negative stain for, because spirochetes are these really, really thin spiral shaped cells. And so if you subject them to heat, they're going to be completely warped and you wouldn't be able to determine morphology or anything like that. So again, you don't heat stain with the negative stain. And this is the stain you use to visualize heat sensitive cells. So remember, safety is going to be on the exam as well. So what's the last thing you should do? You should always wash your hands before you leave the lab. Okay, so real quick, let's remember some PPE. We always have to wear goggles or safety glasses. We provide lab aprons that have to be worn at all times, as well as gloves. And if you have hair that's kind of longer than about shoulder length, give or take, then you want to always pull it back, no dangly jewelry of any kind, and closed foot shoes. Okay, so we now say closed foot shoes because closed-toed, turns out, wasn't detailed enough, because we want the tops of your feet covered as well, right? Because if you spill E. coli, if it gets onto the top of your skin, right, no good. Not the end of the world, but we'd like to avoid it. And so having like a sneaker or a shoe or a boot that completely covers your entire foot is much more ideal. And a lot of flats cover your toes technically, right, but they expose the top of your foot, so we don't want that. And then one of the things you do want to do before you leave, not the very last thing, but one of the important things is clean your workstation. We have Lysol provided in the lab, and you spray down your apron and your workstation with Lysol before you leave. And then says gather your things, and we do not work with our personal objects near us, right? Because if we spill, then not only are we potentially contaminating the workspace, but it could drip onto your backpack or your purse or anything of yours, cell phone. So all of that is put away during lab, and it's actually stored in the back of the room away from any possible contamination. All right, next question, true or false? So the first thing you do before an inoculation is flame your loop. The last thing you do when you're done with an inoculation is flame your loop, okay? You always flame it first because you want to make sure it is sterile before you use it to touch anything, but also because you want to give it time to cool down, because you don't want to flame your loop and stick it straight into any cells because it'll burn them because the loop is hot. So by flaming your loop first and then taking off the lid of your tube and flaming the neck of the tube, right, you're giving that loop a chance to cool down so you don't kill the cells on contact. Remember, the purpose of an inoculation is to grab bacteria on the loop and transfer it into sterile media. And you want to make sure you use aseptic technique so as not to contaminate yourself, your starting culture, or the culture that you're trying to make, or your work environment. All right, next up. All right, so first thing I want to remind you is you can only describe morphology and arrangement if you're looking at individual cells, not a colony, okay? When you're looking at a colony on an agar plate, you use your colony morphology terms for margin and elevation and shape, okay? A colony is a mass of cells. You're looking at millions of cells grown on top of each other, okay? You can see that with the naked eye. You cannot, however, see an individual bacterial cell with the naked eye. That's why we have to stain them and put them under the microscope. So each little bacillus that you see here, that's an individual cell, not a colony, okay? So morphology and arrangement, if you're saying bacillus or cocci, you'd better be talking about an individual cell. You do not use those words when describing a colony. If you're going to tell me staphylo, strepto, diplo, single, whatever, those are for cells, not colonies. Okay, so we have bacillus cells here arranged in very long chains, so that is going to be streptobacillus. Okay, remember, staphylobacillus is not an actual option because they cannot divide that way. All right, 2 more questions. If you want to measure the precise length of a cell, do a negative stain. Yes, yes, and yes. Because remember, no heat fixing during the smear step if you're going to do a negative stain. Okay, because that will warp the shape, which then affects the measurement of the cell. So this negative stain is using nigrosin, which is the acidic negatively charged stain. And so I prefer to use nigrosin over Congo red because it just provides beautiful contrast, right? This nice dark background. You can clearly see these colorless cells. Again, another streptobacillus arrangement in morphology. All right, last question. Streak plate is a quantitative technique. A quantitative technique is where you can determine the actual physical number of individual cells. Okay, so you cannot do that using the streak plate. This is an isolation technique. The goal is to get nice, pretty isolated colonies here in the last part of the plate, and this individual did. Okay, so we can count the colonies, well, for part of it, right? You can't count what's happening in these upper 2 sections, right? There's way too many there. So you can't determine your actual number of cells because you can't actually count any colonies in this area. Okay, so streak plate is not quantitative, but it is an isolation technique. And we use this technique a lot of times if we have a mixed sample. So maybe if you took a water sample somewhere and you wanted to see what was growing, you're gonna have a lot of organisms in that. So it's a mixed sample. And by doing the streak plate, you can look at individual colonies that grow, and each species is gonna grow differently. Okay, so if we're looking at this plate, we know that only one organism grew because all the colonies look the same, right? They're a beigeish round morphology. Now maybe we had red colonies growing and yellow colonies growing. Then you would know the red colonies was a different species, the beige colonies was a second species, and the yellow colonies were a third species. Okay, now it's not as always as obvious when, let me restart that sentence. It's not always that easy to determine how many species are there because a lot of organisms grow as a beige color. Okay, but as you know from looking through the unknown PowerPoints, colonies grow very differently from each other. So even if there are 2 beige colonies, maybe one is a beautiful circle and it has convex elevation, and maybe the other one is a rhizoid and it looks like roots and stuff on there. Well, they look very different. So you would know that those are 2 different species. Okay, all right, that wraps up this review. Please let me know if you have any questions.